Welcome to Microbial Concepts. So this is another video related to interview questions. Okay, but here we are just focusing on pharma or pharmaceutical company related interview questions. Okay, so this is not a video where I am going to discuss all those basic interview questions which are asked to a fresher candidate. Okay, why? Because already separate videos related to these separate topics is already uploaded on my channel. You can find those videos under playlist name Viva Interview Questions. Okay, so you can go there and watch those videos. This video is mainly for, you can say, experienced candidate or those who had worked for say at least 1 year in a pharma industry, so they are expected to know these kind of terms. But yes, there is difference between API and sterile injectables. Okay, so if you had worked in API just say for 1 year, then it is not compulsory that you should know terms related to sterile injections. Okay, so uh, do prepare the way where you have worked. Okay, say if you had worked in sterile injectables, then do focus on sterile injectables and get all the concepts clear. Now here I am not able to cover each and every question, so here I am trying to give you an idea that what kind of questions you may get in an interview, so you can frame questions, okay, related to your topic which you are struggling. So let's start. So types of products the pharma industry produce. They are API, sterile injectables, or the complete drug production. Now it depends in which type of product the pharma industry is producing, where you are working. It depends the questions that you may get. So this video we are going to discuss random questions which are based on microbiology and pharma industry. Okay, so let's start with the fermentation, because let it be a complete drug formation or API or any other kind of drug, fermentation comes into the picture. Okay, so you should know, as a microbiologist, you should know what are the basics of fermentation, what are the parameters which are used to control a fermenter tank. Okay, what are different types of bioreactors, then what are the different parts of fermenters. Okay, all those questions. So let's start. So first, what is fermentation? So it is an enzyme catalyzed metabolic process where organisms, or so you can say microorganisms, they convert starch or sugar to alcohol or an acid anaerobically, releasing the energy. Okay, so fermentation is mainly anaerobically. Then what is importance of fermentation in microbiology? Now fermentation is metabolic process that involves conversion of sugar into other compounds such as alcohol, lactic acid by microorganisms, and it is an important process in microbiology which is used to produce wide range of products. Okay, so you can skip the first sentence and you can say it is a important uh, process from microbiology which is widely used to produce wide range of products, like in case of food, beverages, in pharmaceuticals you can find fermentation technique or technology which is widely used. Then what are bioreactors and types of bioreactors? Now a bioreactor is a vessel like device that provides uniform background for microorganisms to grow and maintain an uninterrupted balance in the biochemical reactions which are carried out by these microorganisms to produce their desired product or desired metabolite. Okay, so fermenter is also known as bioreactor, but when you study the vessel, the fermenter, mainly the term bioreactor is used. Okay, then what are the types of bioreactors? So continuous stir tank bioreactor, bubble column bioreactor, airlift bioreactor, fluidized bed bioreactor, and packed bed bioreactors. Okay, so it is not necessary to give all five types, but try to mention at least three. Okay, that can help you. Then you may get a question like, what is difference between batch, fed batch, continuous process of fermentation? Okay, So here also try to give at least three characteristics on which you can differentiate. Okay, choose any three which are easy for you, like the cultivation system. In batch culture it is closed, in fed batch it is semi-closed, and in continuous culture it is open type. Addition of fresh nutrients is not done in batch culture, and yes, it is done in fed batch and continuous. Okay, then you can say removal of waste. So no waste is removed in batch and fed batch, but yes, waste is removed in continuous culture. So it depends. You can choose any three characteristics and you can give the difference. Then next question is what are the different types of fermentation and how do they differ from one another? Now there are several types of fermentation like alcohol fermentation, lactic acid fermentation, acetic acid fermentation. Now what is the difference? The difference is in terms of the microorganism or the culture that is used and the end products that are produced. Okay. Then yes, a very important question that is mostly asked in the interviews is what is KLA and its methods. So you can say that KLA means oxygen transfer coefficient, which is measured, or it is a measure of efficiency of transfer. Transfer of oxygen between what? Between gas phase and liquid phase. That means liquid phase means your medium, from air to the medium. Okay, in a aeration system. And what are the methods by which you can measure this KLA? So there is dynamic method, static method, off gas method, sodium sulfite method, and dynamic gassing out method. Okay. Yeah, then another part that is very important related to fermentation is different parts of the bioreactors, okay, and their roles. Now, you may not get a question like what are the different parts of bioreactors, but you may get a question directly with the name of a part, that what is the role of, say, sparger in a bioreactor? What is the role of, say, rotameter in a bioreactor? Okay, so go through this table and try to remember in short what are the rules of each and every part of a bioreactor. Okay, so this is also important because you cannot just study fermentation technique without studying a bioreactor. This is the first thing that is taught when you start your uh, by fermentation technique. Okay, then other questions which are related to this topic, that is fermentation, is you can get a question like what is growth curve and what are its different phases. Then what is doubling time, what is passaging, okay, what is seed development. Okay, so you can search these terms on internet, you can search these questions on internet, or even nowadays chat gpt is a very good option to get answers. Okay. Okay, so these are some basic questions which are again related to fermentation. They are what microorganisms are commonly used in industrial fermentation process. Okay, so here the question is what microorganisms. They have not asked you about specific examples. Okay, so you have to say microorganisms which are commonly used are bacteria, yeast, fungi, and then you can give one or two examples like lactic acid bacteria, Saccharomyces cerevisiae, and Aspergillus niger. Okay, then what are some factors that influence the fermentation outcome or fermentation yield? So the factors are temperature, pH, nutrient availability, oxygen level, the type of microorganism that is used. Okay, so it is important to carefully control all these factors to optimize the fermentation outcome. Okay, you need to study all these factors. Okay, what temperature is required for a particular type of microorganism. Okay, not all the time temperature and pH is same in all the fermentation processes. Okay, so it depends. Then how do you monitor and control fermentation process to ensure the product quality and consistency? Okay, this is a question that is likely to be asked in interview because when you're working on a large scale, you need to ensure the product quality all the time and the consistency. Okay, so how you will do that? So by measuring the key parameters. What are the key parameters? We have already seen the factors. Okay, those are the one from which pH, temperature, and nutrient availability are the key parameters. Okay, also you can use techniques to study your fermentation batch. Like you can study the microscopy, genetic analysis can be done, metabolomics to monitor the growth and the activity of the microorganism during the fermentation that is used. Okay, which type of microorganism you are working with, you can study that particular microorganism by uh, doing the genetic analysis, metabolomics to study their growth. Okay, and then you can also use these results of these particular measurements to adjust the process condition or to optimize your fermentation. Okay, if you see that no, the growth, the desired growth is not what you are getting, then you need to adjust some parameters to gain that. Okay, so that's the reason you need to study your fermentation batch. You cannot just, yeah, I have put all everything in the fermenter and I have started the fermentation, so I don't need to look at it. Okay, that's not the way fermentation works. You need to monitor it. Okay, then some questions on sterile water injectables. So what are those? So they are pharmaceutical products uh, that are used for injection. Okay, and it only contains water. Okay, that has already been sterilized and purified according to some specific standards. Okay, so what is the purpose of sterile water injectables? Why we need that? So water is a solvent, we know that. So in medical field, sterile water injectables, they are used as solvent for drugs that are administered by injection as well as for intravenous infusion therapy. Okay, so you need water as solvent for different purposes. Okay, then how was sterile water injectables manufactured? So sterile water injectables are manufactured in very controlled environment using sterile water aseptic manufacturing processes. The water is typically purified using variety of filtration purification method such as reverse osmosis and distillation. Then what is the role of pH in sterile water injectables? Now see, here also you just can't sterilize and purify the water and sell it. Okay, you need to study that as well. So see, here also role of pH is important. The pH of sterile water injectables is important because it can affect the stability and effectiveness of the drug or medication that is been dissolved in it and then administered. Okay, for example, some medications they require a specific pH range to be maintained. Okay, because if not, then they will not remain stable and effective. Okay, so pH is very important for some drugs to remain stable and effective. Then next is what are some key quality control measures that are used to ensure the safety and efficacy of sterile water injectables. So quality control measures, the very first and important, that is testing for microbial contamination and endotoxins. These are very important, and also you have to check the particulate matter if present or not. Then pH, you need to study pH, conductivity as well. Others are like you ensure the water meets specific standards for purity and quality and then properly packaged and stored. Okay, these are key quality control measures to ensure the sterile water injectable that is manufactured is stay safe and it is stored properly and it gives good efficacy as well. Then what are some of the most common challenges associated with the manufacturing of sterile water injectables? So the most common challenges are ensuring the sterility, purity and controlling pH and conductivity of water, and yes, minimizing the risk of microbial contamination during the manufacturing process. Then last, what are the different types of sterile water injectables? So there are several types of sterile water injectables including sterile water for injection, then bacteriostatic water for injection and sterile water for inhalation. Okay, don't get confused, this is sterile water for inhalation. Yeah, so now we are going to see few questions, interview questions related to clean room. Okay, so if you are a experienced candidate, then you know the importance of clean room during the manufacturing or packing process. Okay, so yes, I have included this topic in this video because of its importance, and it is expected from an experienced candidate to know everything related to clean room. Okay, a separate video for clean room and its interview question is again going to be uploaded in few days. Okay, I am working on that, so you will find that video on my channel in next week. And if you are a fresher, then you can learn about the clean room from that particular video, and also you can study for your interview. So what is a clean room? So clean room is a specialized environment designed room to maintain extremely low levels of airborne particles such as dust or microbes or pollens or chemical vapors. Okay, so the air, the main idea is that the air which comes into the room is filtered. Okay, the environment is highly monitored and it is highly controlled. Okay, the humidity of the room, the temperature of the room, the air which flows in the room, everything is controlled. Okay, that's why it is also named as clean room. Okay, now clean room typically uses a combination of air filter, pressure differentials. Okay, even the pressure between two rooms is different, temperature, humidity control, specialized cleaning procedures to maintain the controlled environment. Okay, then what are different types of clean room garments? So there is gowning and degowning process, or say donning and doffing process. So during that, or during entering a clean room, you need to wear few specialized clothings, okay, which are designed specially for clean room, or you can say clean room garments. Why? Because they are designed to minimize particle generation and accumulation in the clean room environment, okay, to minimize contamination, for example. Then they help prevent contamination from clothing fibers, skin flecks from the person who is working, then other sources of particulate matter. What types of clean room garments they are there that we use? So jumpsuit or coveralls, lab coat, goggles, gloves, face mask, hair cap or hoodie, and shoe cover. Okay, these are few to mention. Now it depends on the class of clean room which you are using, you are working. Depending on that, slightly few garments they are or you can see change in the gowning process as well. Sometimes if it is less stringent process, then you can also work on a lab coat wearing the head net cap, goggles, face mask, hand gloves, and your shoe cover. Okay, in case of say you are working in a API or sterile water injectables, so you need to use coverall, okay, and a hoodie properly, so that from head to toe with your shoe covers you are covered. Okay, and two pairs of gloves are used. Okay, so that is a different process overall, and I will be uploading a separate video where you can study how to gown and degown, or how to don and doff. Okay, then what are different classes of clean room and how are they classified? So clean rooms are classified based on maximum allowable concentration of airborne particles per cubic meter of air. Okay, you need to answer this question like it is. The classification is done on maximum allowable concentration of airborne particles per cubic meter of air. Okay, the most commonly used classification system is Federal Standard 209E or 209E, which defines six classes of clean rooms, that is class 1, class 10, class 100, class 1000, class 10,000, and class 100,000. Okay, so this is the classification for clean room. Then what are the sources of contamination in a clean room? So you can answer just like people, equipment, particles which enter the clean room through air, then raw material, packaging, and some manufacturing processes generate particles that can contaminate the clean room. Even this is okay. Or if you have to explain how person or people they are source of contamination, then you can say that people who are working, they shed skin flecks, hair, and clothing fibers. Then in case of equipments, they generate particles through friction or vibration. uh, then through air, particles enter clean room and you need to monitor that. Then raw materials, packaging, supplies, they can also contaminate the area. Then some manufacturing processes, they generate particles or gases that can contaminate the clean room area. Okay, so next is how contamination in clean room can be eliminated or minimized. Okay, this depends on the answer that you give for previous question. You have listed the sources of contamination. Now you can get a counter question that how will you eliminate or minimize contamination in clean room. So proper design and construction to minimize particle entry and provide a controlled environment. Use of HEPA filters, air locks, pressure differentials to control airflow and contamination. Then you can say regular cleaning and maintenance of equipment. Then you can say following SOPs for entering and working in the clean room. Using training, you can train your um, workers. Then gowning degowning process to be followed. Okay, not eating and drinking in the clean room area. Then use of specialized clean room garments while entering. Okay, then regular monitoring and testing of the clean room environment. That means you need to monitor the temperature, humidity, okay, and pressure of your clean room environment to monitor your clean room environment. Then next is what are HEPA filters. So HEPA filters and HVAC, that is heating, ventilation, air conditioning. This is very important because this maintains the airflow. Okay, HEPA filters are high efficacy particulate air filters. They filter the air which comes into the clean room. Okay, so it is designed to remove very small particles from the air. To be classified as HEPA filter, the filter must be able to remove 99.97% of the particles that are of 0.3 microns in size or larger. Okay, so how is the efficacy? 99.97%. Okay, then HVAC uses HEPA filters to filter the air. So it stands for heating, ventilation and air conditioning. It refers to a system. Okay, it is a system which has different parts which work together to give you a controlled environment in clean room. Okay, so it is named as HVAC system. Heating system, cooling system, ventilation system is there. Okay, so it refers to the system that provides indoor environment which is comfortable, and it is done by regulating the temperature, humidity, air quality of a building or a room. Okay, so we have covered sterile water injectables, fermentation and clean room in this video. In another video I will be just focusing on API. Okay, so pharma industry related questions, okay, where we will be just focusing on API. So I hope this video is helpful to you all. So do give a thumbs up, do like my video, do share my videos with your friends and do subscribe to my channel. Thank you.